This is a tale of two oncology research labs with the same goal, but different ways to achieve them. Meet Melissa from Lab A and Philip from Lab B. Though they are hundreds of miles apart and face different challenges, they are on the same metaphorical path towards discovery. Both labs are attempting to discover new therapeutic targets for personalized medicine by characterizing target proteins or discovering new biomarkers. It's a pretty daunting task. The human genome is estimated to contain 20 to 25,000 genes, but due to regulatory steps like post-translational processing and modification, the human proteome contains more than 1 million proteoforms. Dealing with that complexity is a roadblock that Philip and Melissa have in common. And although their testing methods differ, they both struggle with the immense amounts of time and data required to discover novel drug targets and biomarkers. For Melissa and Philip, discovery research is like looking for a needle in a haystack. So the roadblocks they both face on their path are like giant hay bales blocking the way. Melissa's lab uses trusted methods like Western blots, flow cytometry, and IHC that are slow and steady. She and her team essentially pick through giant bales of proteomic hay one by one, looking for the right needle. It's a process that works, but she has more hay obstructing her path than Philip. And because of the tedious nature of the work, she's in a constant race against the clock. Philip's lab, on the other hand, relies on liquid chromatography mass spectrometry, or LCMS, for proteomic screening, which produces boatloads of data points to sift through. It's sort of like using a hay processor that spits out endless strings of hay data noodles. Philip was able to bypass much of the manual hay picking done by Melissa's lab, only now he's got hundreds of boxes of hay to sort through. So although Philip is already halfway to the solution, he's still facing a problem. Both of our lab managers ask themselves, how can we find the needles quicker and without generating impenetrable amounts of data? The answer? With a magnet. With PTMScan from Cell Signaling Technology, you enrich samples before LCMS to highlight precisely the candidates you want for further analysis. It's like holding a powerful magnet over your haystacks, specially tuned for the right targets, post-translational modifications or post-translationally modified peptides, so you only pull out the needles you need without all of the hay or background noise, all at once. Essentially, the power of PTMScan is simplification. By enriching with the right PTM motif antibodies, you decrease the complexity of what goes into the instrument. When you decrease the complexity of the sample, the instrument spends more time focusing on what you're interested in, increasing the likelihood that you'll find what you're looking for. Instead of thousands of hours it takes generating small amounts of data at a time, using traditional biochemistry, PTMScan makes the switch to mass spectrometry, LCMS, incredibly simple. PTMScan allows you to look at hundreds to thousands of proteins and their modification sites, PTMs, in any number of samples and quantify differences between samples. And these are all valuable data points. But the quality of the data isn't the only value. It's also in the time and energy saved getting there. Instead of months spent picking through hay, you can have all the right needles in far less time. For Melissa and Philip, the implications of that efficiency could mean serious breakthroughs. That's precisely why the scientists at Cell Signaling Technology developed PTMScan. As a company rooted in science, our goal is to further the potential of proteomic testing and enable labs and lab managers like Melissa and Philip to make life-saving discoveries. You can access the power of PTMScan through our products or services. With our kits, you can perform enrichment, LCMS, and data analysis in your own lab. With our services, all you need to do is send us a sample and we send back the data. Philip was already using LCMS in his lab, so the kits were a perfect fit. Melissa wasn't able to run the analysis in her own lab, so she took advantage of the services and let CST generate the data and even helped her analyze it. Armed with PTMScan technology, Philip and Melissa now have a clear path to discovery. To find out more about how PTMScan can simplify your proteomics work and change the way you think about discovery, click here.